Thank you so much, Eddie. Okay, today I would like to talk on marine science with AI for sustainability and well-being. Actually, a little bit I like to focus on marine genomics or marine metagenomics. First of all, I would like to thank the Holy See and the President von Braun and the Cardinal Taxon and all council members. Thank you so much for giving me such opportunity. Actually, as you may know, the United Nations declared the starting of United Nations Decade of Ocean Science for Sustainable Development, starting from 2021 to 2030. So just this new project started under United Nations program. And before that, like Tala Foundation and the EMBL conducted so-called Tala Ocean exploration and sailing over the world, sampling the seawater, then check all data, biochemical, physical data, and also DNAs. This is Spanish Malaspina expedition, like 2010, almost more than 10 years ago. Already, also, they did the similar thing. Actually, I started marine metagenomics in 2005, collaboration with Fishery Research Agency in Japan. We have not sailed over the world, but mostly near Japan island. So metagenomics, and particularly one of, I would say, field will be marine metagenomics. What is metagenomics? Actually, it started like 2004, showing that without identifying any biological identity, just you can sequence a group of sequencing, that collection of fragmental genome. Then just you sequence it, then after that, you just check with the database. Then you know what they are, or what you don't know. In the case of the seawater, we just take a seawater, then filtering. Then from remaining things, we isolate DNAs, immediately sequencing. Then we check with a database. There are two ways. One, amplicon-oriented metagenomics. The other one is totally random shotgun metagenomics. Now, random shotgun method would be mainstream right now. So just like those Lego pieces and a bunch of different fragments. And currently, the filtering missing viruses, marine viruses, usually it is said that more than 30% of microbes in a marine would be viruses, but we are missing that. Then after that, we call annotation or classification. We have to make this kind of things. What kind of organisms or what kind of genes? Then we construct database, and should be integrated database. Not only DNA information, but like sea regions or seawater temperatures or current kind of things. And we published one book. I think more than 10 years ago, I moved to Saudi Arabia. Then I started metagenomics in the Red Sea. Why Red Sea? Red Sea has high temperature. It is known to be a model after climate change because no inflow rivers and very high temperature, and we conducted metagenomics. Then I don't have enough time to show that, but we started to understand how many genes they are in the ocean. We call KAUST Global Ocean Genome Project. Then even the ocean, it's huge, but still we decided not ocean. We should include other domain, biofields, to understand ocean meaning. And collaboration with my colleague, Carlos Duarte, others, and we conducted the project. And this is because, as Professor Eric Randall explained, huge reduction of sequencing cost. So you can just sequence it. Instead, computation, heavy computation is necessary. Of course, AI would be a key issue. Then we constructed metagenomic analysis platform, KMAP. And this is an annotation pipeline. It's a very tedious, but still it's so much bioinformatics tools and the platform needed. This is a kind of current, I would say, outcome, and you cannot see. However, most importantly, what I like to mention is here. I think we collected 1 billion redundant genes. When I say genes, the definition of genes is complicated. We focused on only protein coding regions. Then now, 0.6 non-redundant genes in many different biofields. What I'd like to emphasize, if you take the number of genes over the time, never saturation, still it's increasing. Of course, from here, such a huge data set can be utilized for digging the gold. We call blue gold. You can see bioenergy or detergent for lipase, or like any other utilized genes or gene set in even antibiotics. So almost 18 years ago, we published one opinion paper in Nature, the big data. The life science will be faced by big data. Actually, we know from human genome, we already that. But beyond that, we have data is coming. And we call it genomic Big Bang. So in Taiwan, we organized international conference stating genomic Big Bang. 2 implications. One, currently such a Big Bang data is coming. And later on, I would say, what would be origin of genome? There could be Big Bang. And of course, AI would be a key. So in Japan, Marine Open Innovation Project Institute was established. I'm now pushing this one. And this is more like biotech and looking for useful genes or gene product. But then, more importantly, what is a genome zero? And what is origin of genome? Of course, the origin of life is very important. But LUCA would be, it's really only one. If you define ancestral genome as the most synonymous gene set, it's enormous. You cannot have only one single cell because more number of genes coming. Then one possible implication would be, I think, ancestral genome not to be single, only one, maybe many different sets. Of course, it may not be consistent with origin of life. Origin of life could be one, we don't know, but the genome would be, it cannot be explained. Only a single genome can develop to the present. because we don't know how many genes on the Earth we have. It's a very serious, important issue. And now NOAA started observation, not using exploration ship, more like a AUV, autonomous undersea vehicle, namely seawater drones. They are putting along the ocean. And then space would be a key. I think those data will be collected through ocean, space. And then those kind of like a Starlink and this kind of space structure will be strongly connected to ocean science, particularly genomics. And how we can utilize AI. But currently, still, I think AI is under development. Particularly, not only genome, there are different kind of data, seawater, or even depths of levels. And then we propose kind of a dot digital data DNA chip. And those like DNA's fragment can be mapped on the virtual chip on the computer. We can have some pattern. So this can be utilized not only DNAs, but also like environmental data. So those the chip pattern recognition will be easier by utilizing AI. And the integrated data analysis will be possible by conducting such kind of digital dot presentation. So like different locations, different depths, and different features, and all can be represented by dot pattern. Those dot pattern can be integrated by AI because pattern recognition would be very good at AI. So therefore, then those data can be shared. And as I said, the space. So like a Starlink, all space observe those AUVs and collecting the data. Right now, NOAA does not have a DNA sequencing capability of AUV, but still it will be possible like a nanopore technologies or something. Such a space and the ocean develop ocean science, particularly ocean genomics, enormously. Then we have to have a data policy up to how you can exchange the data, and up to what you can have intellectual property. What would be like national security? Because those we should have sound sensor, sonars. So it's a totally difficult, but a very interesting future is coming. Therefore, we have to prepare for that. So thank you so much. This is my conclusion and this acknowledgement. Always DNA should be for world peace. Thank you.